Bismillahirrahmanirrahim. Assalamu alaikum. I, Dr. Hooria Aamir, Head of Physiology Department, welcomes all my students of MBBS and BDS to today's lecture of the cell membrane bounded organelles. And what up till now we have done about the rest of the organelles in the previous lecture, you are very much familiar to what is endoplasmic reticulum, what are its types, what are lysosomes, what is Golgi apparatus, what is the process of phagocytosis, what is pinocytosis. Now today's lecture is actually going to encompass the rest of the cell organelles, and one of the very important one is peroxisomes. Now, it was discovered by a Belgian scientist in 1967. It is a membranous bound organelle. When we are using the term membranous, it is actually composed of cell membrane sort of structure. Now, it is about about half or one third of the size of lysosome, much lesser in size in comparison to lysosomes, and there is presence of several hundreds of these cells, these type of organelles in all the cells, and it is produced by the endoplasmic reticulum, not from the Golgi apparatus. As Golgi apparatus, they are producing lysosome, the endoplasmic reticulum by the process of budding off or by the process of self-replication, the peroxisomes they are being produced. Now, there is presence of different type of enzymes given the name of peroxides, or oxides or peroxins. 32 types of proteins they are being present, and they are containing the oxidases in it, the oxidative enzymes which use oxygen to strip off hydrogen from organic molecules. Hydrogen peroxide H2O2 is formed, which is converted into water and oxygen by the catalase present in peroxisomes. Beta oxidation of long chain fatty acids in liver cells also occurs, and it detoxifies various wastes, especially in liver cell. And if it is absent, God forbid, then one of the diseases can be like Zellweger syndrome. So you should be knowing about the name of Zellweger syndrome. It is quite frequently asked that how you can compare and contrast between lysosomes and peroxisomes. Always make a good list of comparative points in your mind when you are preparing for your viva or your theory exam. Now, see how the endoplasmic reticulum is going to give rise by the process of budding off to the vesicle, which is the precursor of the peroxisome. Later on, the specific proteins they are being imported into this vesicle, giving this vesicle the new name, and that is the peroxisome. And then peroxisome later on can be, by the process of fission, can be divided into two later on. But the original precursor is always the endoplasmic reticulum, and its growth is by the uptake of specific peroxisomal proteins and lipids from the cytosol. This is the process by which the new peroxisomes can arise. Now, when we are doing and learning a lot about the different organelles, we should be knowing about the cytoskeleton. What is the cytoskeleton? Cytoskeleton is actually the protein scaffolding in the cytoplasm, which is providing the support to organize, anchor, and control the movements. So, the cytoskeleton is composed of microtubules. Microtubules, they are like a nanometer size in diameter. They are the long, hollow, unbranched tubes composed of alpha and beta tubulins. There is presence of 13 units which are present in the one turn of spiral. They are essential for positioning of organelles, and they maintain the shape of cells and nerves. They are actually providing the proper formation and shaping of the cells, the nerves, and so. See, these are the microtubules. actin filaments, the intermediate filaments. See how this microfilament and intermediate filaments, they are providing the different anchoring support formation to particular cell. They are responsible for making the basic architecture of the cell. See the tubulin protein arrange in a helix to form 13 vertical filaments around a hollow core. Complex cell movements. Now, how the complex cell movements they are taking place? There is presence of the vesicular transport, the molecular motors, the protein attached to vesicles and use the ATP to walk along. There is presence of protein like kinase and dynein. Herpes, polio, rabies virus use this route, and movements of cilia and flagella. Cilia, they are short, tiny hair-like projections, and they are present in the respiratory tract, oviduct, and ventricles, and they are responsible for movements and guiding. The primary cilium, the sense regulatory signals like the growth, differentiation, proliferation, the sperm cells, they do have flagellum. Then there's 9 fused pairs in outer ring and the 2 single unfused in the center. The diseases may occur if there is immotile cilia syndrome. C. Outer microtubule doublet, then dynein arms, then central microtubule, there is radial spoop. C. Microfilaments. They are small, 6 to 9 nanometers. They are globular subunits organized into double helix to form the strands. And in muscles, they are organized along with the myosin to give rise to the muscle structure. And under membrane, um, they are going to give support, and they do change their shape during the course of endocytosis, locomotion, and important for the cytoplasmic streaming, cytokinases, responsible for amoeboid movement, assembly and disassembly in sperm cells, white blood cells, fibroblasts, and skin cells. Then there is presence of intermediate filaments. They are 7 to 11 nanometers in diameter, irregular thread-like molecules, tough, durable fibers being stabilized by the lateral bondings, resistant to external pressures, keratin in epithelial cells, nails, hair, horns, feathers, scales, neurofilaments. They give strength and stabilize the nerve cells. Um, so this is all about the different aspects regarding the cell physiology. Now you are all able to know the structure of the cell and how it is working, how the organelles they look like and how they are functioning, what are the different membrane bounded organelles, what are the non-membranous organelles. So you're all supposed to give a good reading from Guyton Physiology and do the portion in your workbooks so that this topic should be with you forever, homeostasis, and it is a sort of open chance for you all to get very good marks in your written as well as for the Viva examination, because all the things they are there which are very important like homeostasis, the feedback mechanisms, the internal environment, the concept of this smiley you interior, you all know up till now, and you're all supposed to be able to give the examples of homeostasis in your body, like what is the feed forward mechanism, what is negative feedback mechanism, what is positive feedback mechanism. You should all be able to do it. So that's all for today's lecture, and stay blessed. Allah Hafiz.